Hello viewers, in this particular lecture on food microbiology, I shall be introducing the different aspects of food microbiology. Food microbiology actually include processing of food, preservation or canning, fermentation, and of course the different fermented end products which include the different secondary metabolite, and also I shall end up this lecture with the different foodborne pathogen and spoilage of food. To start with, the food processing, which include the different aspects of cooking or modifying food, which actually started way back in the prehistoric times, when human beings first learned the use of burning the food, or they call it proper roasting of food. And this roasting actually resulted in the burnt protein and it gave some flavor. So that was the first incidence of food processing. Of course, now there are different mechanisms by which food is processed, and at the same time when it is processed, the microorganisms are removed from it. So the most important thing is that how to preserve the food, and that naturally include food preservation. The history of food preservation is dated back to 1809, when this gentleman Nicolas Appert in 1809 actually he was working for the French troop, and he discovered a unique bottle with a rim which was actually a container where processed food or food could be stored for a longer period. And subsequently, this started the mechanism of food packaging, and nowadays we have packaged food where the primary criterion is that there should not be any toxin present inside the packaged food, as this particular packaged food contains contaminated fungi or mycelial fungi or different types of penicillium or aspergillus or mold fungi. And of course, when the food is packaged, or even if it is not packaged properly, it should be kept in low temperature condition, and that means refrigerated food transport as it is done in this modern refrigerated van. But even in the refrigerated condition, there may be different microorganisms growing. So these are refrigerated meat where there are trace amount of salmonella growing. As when seen under electron microscope, these are the meat fibers where these rod-shaped bacteria have grown and contaminated the meat products. And mind you, these particular contaminating organisms grow at a very low temperature, maybe around 4 degrees centigrade, which is the normal refrigeration temperature. And this type of salmonella silently get into our body through our digestive tract, and it can cause severe symptoms which is coming under the disease of salmonellosis, which include nausea, vomiting, diarrhea, and severe abdominal cramp. And it can be sometimes very severe, which can cause actual damage to the digestive tract. And of course, the best treatment is antibiotic therapy, which can be either by oral route or injectable. So naturally, present-day food should be stored in the proper way. And what are the parameters of storing? They should be stored in nice tin container. And this tin container actually should be there after the food has been processed. So during processing, we should minimize the use of human agency or human hand. So this type of mechanized peeler can be a very effective tool by which we can reduce human intervention, and thereby we can prevent the bacterial contamination. Of course, frozen condition. In the frozen condition, a food can be stored for a long period, and suppose these are the pea, which are this is the stored condition in refrigerated condition, and then after soaking them in water, they are coming back to their normal state, which can be used for consumption. And this type of pea can be stored for a long period, as it is observed in a steamship called Bertrand, which was crashed, and after crashing, after nearly 30 years, these containers were recovered, and it was found that the peas were absolutely normal. So this type of canning and freezing the food can keep them for a prolonged period without bacterial contamination. So this particular phenomenon by which the food can be stored in a particular container without contamination was denoted by Peter Durand, and he actually showed in 1810 that a particular tin container like these containers, if the food is stored, he called them double seam container, it can be protected for a long time against bacterial contamination. So this is the typical cylindrical double seam can, and the steps of making this double seam can include the rectangular pieces are cutting out or cut out, and then the rollers are used, and then they with a die cutter typically circular pieces of this metal were added and they were actually sealed on both sides. So that actually make a perfect sealed container. But coming to the next important aspect, which is the phenomenon of pasteurization, that is, before the food is stored, it should be at least free from contaminating bacteria, at least the vegetative forms. And it was given by none other than Lazzaro Spallanzani, who actually for the first time in 1768 denoted the phenomenon of high temperature sterilization with a very simple experiment. There was some gravy, and one gravy was boiled while the other one was also boiled, but in one case it was sealed, in the other case it was not sealed. It was found out that the gravy finally was having maximum amount of microbial growth in that case where there was no cover, but in the other case where there was the boiling followed by sealing, there was no contaminating bacteria or contaminating microbe. What does it indicate? It indicates that once the food is boiled and stored in a microbe-free condition, it can be maintained for a long period. So this started the idea of pasteurization. Of course, it was Louis Pasteur who in 1864 actually for the first time showed the phenomenon of wine pasteurization. And these are the big barrels where the pasteurization were carried out and stored, and the wine remained for a long period, that it had a very long shelf life. But much later, it was Soxhlet in 1886 who actually started this unique phenomenon in case of milk. So milk pasteurization is a technique which is even now followed. And this type of milk pasteurization ideally can be high temperature, short term. That means the milk is heated to 72 degrees centigrade for around 15 seconds and then it was cooled. And this particular milk is free from all the vegetative forms. But mind you, there will be the spore forms which will be germinating and will spoiling the milk after 48 hours if it is not boiled properly. There was another method which is called ultra high temperature sterilization. What is the phenomenon? In this particular phenomenon, actually the milk is heated to 140 degrees centigrade for 2 seconds and effectively it was immediately packaged and there was no contaminating bacteria. In US, this type of method was followed. Instead of this 65 to 70 degrees centigrade, they are using the double temperature and they are reducing the duration of boiling. But at the same time, it resulted in long-term preservation of the food, like we get effectively increased shelf life milk, we call it ESL, or this particular enhanced shelf life milk are very common in the Western world, though in order to make the milk free of contaminant, there are some preservatives added to this particular milk. But of course, when I say that the food is being contaminated by microbes, sometimes the microbes also can be used to reduce the growth of other microbes. And there are different bacteria which produce protein which are called bacteriocin or bacteriolysine, like the nicene, which is using simple lipid port, lipid 1 or lipid 2 port to enter and to destroy other bacteria. And sometimes this bacteriocin can directly penetrate through the membrane of the bacteria and destroying the bacteria. So by growing one type of bacteria, you can actually get rid of another type. So we can call it biological control, but in the way this type of phenomenon is helping us in keeping the food bacteria free for a longer period. Apart from using bacteriolysine, bacteria can also be controlled by another group of organism, which is actually a virus commonly called bacteriophage. These bacteriophage can straight away bind to the cell wall of the bacteria and their DNA will enter the bacterial cell and will cause the lysis of the bacterial cell. So if the bacteriophage is present in the food material, they also can control the growth of bacteria in that particular environment. Of course, sometimes it so happens that bacteria will produce some metabolic products like the organic acid, which will make the pH acidic, and thereby they themselves can control their own growth, or rather I can say by the modification of the pH or the lowering of pH, we will also check the bacterial growth in a particular food material. The next important aspect is the product of different fermentation of different microorganisms. And one such important fermentation product is ethyl alcohol. Ethyl alcohol mostly is being produced by a unicellular fungi or the yeast. The Saccharomyces cerevisiae, which is converting glucose to pyruvic acid, then pyruvic acid is converted to acetaldehyde, and finally ethanol is produced. So alcohol production is a very important fermentation product, and commercially it is being utilized where there are multiple such container where the medium is added. To this particular medium, the yeast inoculum is added, then the fermentation is carried out, and after fermentation, filtration is carried out, and finally the end product ethanol is obtained. Preferably, most of the commercial strains of yeast can produce ethanol up to 15%, and this 15% is the commercial variety, after which distillation is carried out for the purification of ethanol. But beyond 15%, very few strains of yeast can actually tolerate this particular alcohol percentage. Apart from ethyl alcohol, bacteria can also produce lactic acid. In fact, it is glucose which is converted to pyruvic acid and it is converted to ethanol by alcohol fermentation, and at the same time there will be some bacteria which will be converting that particular pyruvic acid to lactic acid, and it will of course utilize the energy of reduced NAD. So lactic acid and alcohol fermentation together will produce heterolactic fermentation, and pure lactic acid is produced by lactic fermentation. Apart from alcohol or lactic acid, the microbes can produce different other type of polysaccharide, and one such particular algae, which is a brown algae, Laminaria hyperborea, is producing a particular polysaccharide called alginic acid or alginate, which is a polymer containing the beta-1,4 glycoside linkage linking the D-manuronic acid and the L-gluuronic acid. So it is a polysaccharide which is having use in medicine as well as in different other purpose. There may be organisms which will produce cellulose, which are just the beta-1,4 glycoside linkage of glucose residue. So it is also called the glucan. In the same way, they can produce the polygamma glutamic acid, which is also industrially important, produced by different bacteria like the bacillus. Most importantly, bacteria, they do produce outside their cell wall, which is called extracellular polysaccharide, EPS, and this EPS production can be utilized in the commercial means or in the production of different medicine, and they are commonly the dextran, which is deposited outside. Now, why these microbes produce EPS? In order to have some connection with each other, or we call it in the technical terms quorum sensing. Now we come to the most important aspect of food spoilage. Different fungi, different bacteria can actually bring about the spoilage of food. Like the common bread, these green colonies of mould, which is mostly aspergillus and penicillium, can spoil the bread and can produce toxin in it. But when we store the pea and yeast is growing on it, then there will be this brown slimy growth on the pea which will result in the destruction of the pea. At the same time, Botrytis cinerea, which is a fungi, actually is sporulating and growing on grapes and strawberry, causing their destruction. At the same time, there will be penicillium rot, which is an opportunistic pathogen spoiling the tomato. There will be the Guignardia bidwalli causing the black rot of grape, and there will be the Sclerotinia rot of apple. All these diseases actually can destroy the food material, including the different fruits, reducing their shelf life. Not only fungi, there may be some bacteria causing the soft rot, or the Arvinia carotovora causing the soft rot of tomato, which is actually producing an enzyme polygalactouronase, and which will destroy the outer skin of the tomato and making it rot at a quicker time. So it will reduce the shelf life of tomato. Now we come to the different foodborne pathogen. What are the different pathogens that can enter through our food? It includes the different enteric virus like the hepatitis A virus causing jaundice, a rotavirus causing severe dysentery or diarrhea, and astrovirus also doing the same. These are the electron microscopic figure of these type of viruses entering through contaminated food and water. There may be the waterborne cyst of Giardia. When they enter, they become active, and these active trophozoites cause the infection. We call it giardiasis, which can be very common in children because they normally take anything and everything they come across. So naturally they are particularly suffering from giardiasis, which is actually a stubborn type of infection in children. The next group is the fungi. The fungi, when growing on different food material, can produce different toxin. One such toxin is the argotamine, which is produced by the argot fungus or Claviceps purpurea, producing this argotamine, which is causing the contraction of the smooth muscle of the uterus, which may cause abortion in pregnant women as described in cattle. Or there may be this Aspergillus flavus, producing the dreadful aflatoxin B1 or other derivatives of G1, G2, etc., causing the severe problem of cancer. So Aspergillus toxin or aflatoxin is a potent carcinogen coming through foodborne sources. Coming to some bacteria-borne pathogen or bacterial pathogen, which include the Staphylococcus aureus, causing the skin and soft tissue infection. There may be the Yersinia enterolytica, causing the enteric infection or diarrhea-like symptom. These are all same study of the same bacteria. Or there may be the different Vibrio contamination causing cholera and also causing hemolysis. So very few people know that there may be the Vibrio parahemolyticus producing the dreadful protein of hemolysin, which is degrading blood or causing hemolysis. There may be the Campylobacter jejuni, which is also foodborne, which can tolerate low pH, and they can cause the severe Guillain-Barré syndrome. What is that? The nerves are damaged. The nerves are so important because it is carrying the stimuli to the muscles. So this particular neurotransmission is blocked because the nerves are getting damaged because their myelin sheath are destroyed by the bacteria, and this is commonly known as the Guillain-Barré syndrome. There may be the Listeria monocytogenes causing the disease of listeriasis, or the Salmonella infection causing the disease of salmonellosis, which can invade multiple organs, but primarily the source is the digestive tract or the alimentary canal. The shigellosis, which is another dreadful infection which comes with amoebiasis, is caused by Shigella. And the best way to detect is the Shigella presence in the stool sample. Or there may be E. coli infection. E. coli can be of various types. E. coli is a very common bacteria which is present in our small intestine, but it can be enterohemorrhagic, it can be causing hemorrhage in our intestine, it can be enterotoxigenic, producing toxin, it can be enteropathogenic or enteroaggregative, which is causing the traveler's diarrhea or persistent diarrhea, or it can be enteroinvasive, that is, it is invading other organs from the intestine. And finally, it can be diffused. That means it will spread along the entire intestine, causing the infection of diarrhea or making the situation worse. This is the endospore of Clostridium botulinum. So even we can boil the food, we can actually make the food pasteurized, but these endospores are not destroyed. So this type of Clostridium botulinum endospore will actually cause the disease of botulism, which is a very bad type of disease because it actually is preventing the neurotransmission and contraction of muscle. And one such important toxin is the Bt toxin produced by the Bacillus thuringiensis. And this Bacillus thuringiensis will also block the acetylcholine, and thereby the neurotransmission is blocked to the muscles and the muscle contraction is prevented. And this type of Bt toxin can also come through food. Coming to another very important bacteria, which is the Bacillus cereus, which is actually a common soil-borne bacteria which produces toxin, which is a 3-component protein toxin or the CYP toxin. This CYP toxin is an emetic toxin which is blocking the neurotransmission and also digestive phenomenon and the defense response. In severe condition, it can induce death. So in this particular lecture of Introduction to Food Microbiology, we have learned the different aspects of food microbiology, that is processing of food, preservation of food, fermentation of food, and at the same time I have concluded with the different types of foodborne pathogen and the pathogenesis that they are causing in human beings. Thank you.